Chapter 7. The Origin of Species discusses how different species can be identified or differentiated and how these differentiations occur over time. It'll also discuss some of the factors that can contribute to speciation and some of the history behind what we know about speciation today. Ernst Mayr was a German zoologist who explored the New Guinea forest, searching for new bird and plant species. His expedition into the forest was backed by Lord Walter Rothschild. In the end, he brought back 26 new species of birds and also 38 species of orchid flowers. In 1963, he published Animal Species and Evolution, and is considered one of the leading evolutionary biologists of the 20th century. His contributions played a crucial role in bridging the gap between Mendelian genetics and Darwinian evolution. This unification was called modern evolutionary synthesis. Mystery of speciation. Through various observations and studies, evolutionary biologists have noticed that nature is discontinuous, but species are discrete and remain in clusters. The question here now is, how can different species be discrete and discontinuous when evolution is continuous? For example, the varying images that come to mind when you hear wildcat. This idea was also tagged as the mystery of mysteries. This observation would also lead us to believe that how new traits arise is just as important as how new species arise as well. Species identification. The morphological species concept is defined as a group of individuals that resemble each other more than they resemble members of other groups. One problem that should be noted here are species that display sexual dimorphisms. Sexual dimorphisms are differences in, say, behavioral or physical traits between the two genders of the species. A new question that arises then would be, how do some species that are almost identical still be considered different species? The answer would lie in how the seemingly similar species interact, or if they interact at all. This would mean that species that are similar may share the same area but never exchange genes as well. The example here shows the various species of Drosophila, or what they are better known as, fruit flies. Though the different species are very similar and sometimes unidentifiable from each other, they still maintain their speciation. But why? Similar but different. Members of different species may be able to coexist, but may also be reproductively isolated from each other when they do not share the same gene pool. This gives way to a new concept, called the biological species concept, which is defined as a group of interbreeding natural populations that are reproductively isolated from other such groups. This was defined by our friend Mayr in 1942, and now serves as one of the main identification tools in new species discoveries. Some of the reasons for why species may become reproductively isolated can be, for example, different mating seasons or pheromones, or the more common geography as well. The picture here describes how a species of lizards diverged into multiple species across California. Most of the different species you see here scattered are most likely due to geographic reasons, as stated in the slide. Biological species concept. From the definition of the biological species concept mentioned earlier, this would mean that a species is then an evolutionary community. For example, mutations can arise in a community or species, but they will most likely not be able to reach another biological species, even if they are similar. This can be a reason for how some populations diverge and become their own species. One red flag for the biological species concept, though, would be for a species that go extinct, as they must then be categorized using the morphological species concept, as their behaviors and interactions cannot be observed for obvious reasons. A majority of speciation can be attributed to geographical isolation. The picture here shows an example, though probably uncommon, of a species of frogs that may speciate with the formation of a road between them. Conditions on either side may be different, thus creating a different force of natural selection for both new populations, resulting in possibly two new species.
There are four predictions that must hold true to support the theory of speciation by geographic isolation. First prediction: if speciation depends largely on geographical isolation, there must have been lots of opportunities during the history of life for populations to experience that isolation. Geographic isolation is common, and it can be caused by mountain ranges rising, glaciers spreading, deserts forming, and so on. Each event brings a new chance for a species to be sundered into two or more populations. Even a river can act as a geographical barrier for bird species that don't like to fly over water. Another way for populations to become isolated is through accidental long-distance dispersal, where a few individuals colonize a distant land. Second prediction: if populations must usually be physically isolated from one another to become species, then we should find the most recently formed species in different but nearby areas. Time can be estimated based on the differences in their DNA sequences. Thus, most similarity in DNA means that they are sister species, and the population split was the most recent. We can see this in nature. Each side of the Isthmus of the Panama has 7 species of snapping shrimp in shallow waters, with the closest relative of each species being on the other side. There must have been 7 ancestral species that were divided when the isthmus arose 3 million years ago. Third prediction: geographic isolation is still occurring in nature. This is confirmed because we can see different cases in which two isolated populations of a single species show different stages of reproductive barriers. Fourth prediction: reproductive isolation between a pair of physically isolated populations increases slowly with time. This is tested by observing many pairs of Drosophila species that had diverged at different times. Divergence time had been estimated based on differences in DNA sequence. Results showed 3 levels of reproductive barriers that arose with time: mating discrimination between the pairs, sterility of hybrids, and inviability of hybrids. Sympatric speciation. This form of speciation does not require physical isolation, but is uncommon because interbreeding will pull populations back towards each other. Best place to look for evidence is small habitats in which there are no barriers that could have isolated the closely related species. The best example for sympatric speciation is cichlid fish in the 2 tiny lakes in Cameroon. The lakes are too small for species to become physically isolated, but yet multiple related species can be observed in each. Special forms of sympatric speciation. Allopolyploid speciation starts with the hybridization of 2 species that live in the same area, but it requires 2 things: that the parent species have different number or types of chromosomes, and that each set of chromosomes must be doubled. Hybrids would be interfertile with other hybrids, but not with either of the parental species. Autopolyploidy is another form of polyploidy that can arise simply by doubling all of the chromosomes of a single species. They are able to produce fertile offspring with other autopolyploids, but only sterile offspring with the parental species. Polyploidy requires the union of sperm and egg with abnormally high number of chromosomes in 2 successive generations, and it is much more common in plants than animals.